Hello, good morning everybody. This is your lecturer D. Rajashekar, Department of Botany, Srimati Ganganeni Kalyani Government Degree College, Pinikonda, Guntur district. In this video lecture, you are going to learn what is a cell theory, differences between prokaryotic and eukaryotic cells. We all know that all the organisms are made up of cells, but there are certain organisms they have only one cell, that is the organism and that is the cell also. The cell is a structural and functional unit of an organism. An organism is formed or constructed by cells, and the functional aspects, all physiological aspects, are done by cell. The cell is a structural and functional unit of an organism. First of all, the scientists named Schleiden and Schwann, they belonged to Germany, proposed a primary theory about cell in 1838. According to them, cell is the basic structural and functional unit of an organism, and they also proposed a cell theory or cell principle. Schleiden also identified nucleolus present in the nucleus for the first time. Later, in 1846 and 1855, the two other scientists by name Nageli and Rudolf Virchow said that all the cells do not get formed on their own, but they are formed from the pre-existing cells. And they said omnis cellulae cellula, that means the cells come from previous cells. And basing upon all these observations made by all these scientists, few things have been considered, and they are called cell theory. What are they? Actually, all the organisms are made up of small cells. All the metabolic activities happen in the cells. They do not form spontaneously, but they form from pre-existing cells. That the cells are carried to many generations continuously. These are some of the important points of cell theory. And later, even before this, actually, a scientist, Robert Hooke, identified cells in the cork of plants. That is a dead cell actually, but later Anton Leeuwenhoek also observed bacteria, a living cell, for the first time. Those cells seem alike, but there are certain differences in the cells. Morphological, physiological, anatomical differences we can see. The cells are basically classified into two types. They are prokaryotic cells and eukaryotic cells. In this slide you can see prokaryotic cell. Pro means primary and karyotic means carriers and means nucleus. The cells with the primitive or immature nucleus are called prokaryotic cells. The best example for this is bacterial cell. Here in this photograph you can clearly see it is a capsule-like structure enclosed by cell wall, and above the cell wall you can see a capsule. Below the cell wall you can see plasma membrane, and on the surface of cell you can see small hair-like appendages called pili, and a long whip-like structure, flagellum. And inside the cell you can see a tangled mass, that is DNA, and it is also called nucleoid. Why it is called nucleoid? Because it is not enclosed by a membrane. That is why the nuclear material is seen dispersed in the cell. Such primitive nucleus is called nucleoid. And inside you can see blue color dot like structures. They represent what? Ribosomes. Thus, prokaryotic cell shows very few cell content like nucleoid, cytoplasm, ribosomes, pili, flagellum, cell wall, capsule, and membrane. We cannot see any major cell organelles in them like chloroplast, mitochondria, endoplasmic reticulum, ribosomes. They cannot be seen here. So the prokaryotic cell is with very few cell components. If you observe eukaryotic cell, the best example for eukaryotic cell is a plant cell and an animal cell. Here we can see a figure of plant cell where it shows cell wall. Below the cell wall you can see plasma membrane. In animal cell wall is absent. Below the plasma membrane we can see cytoplasm. In the cytoplasm we can see chloroplast, which is meant for carrying photosynthesis, mitochondria for respiration, ATS type of ribosomes, Golgi complex, smooth-walled and rough-walled endoplasmic reticulum, and you can see a clear nucleus close to the endoplasmic reticulum and all other microbodies. And you can also see a huge vacuole which is called cell repository, because all the waste materials are dumped in this vacuole. Thus it seems very clear, and with all the components like all cell organelles, important cell organelles like nucleus, mitochondria, chloroplast, and all that, and so that it seems it can carry out its own activities even outside the organism, that it is fully established, thus it is very fully developed. Whereas in prokaryotes it is with very few components. So after seeing the two photographs and the very basic details, now we'll focus on the differences between prokaryotic cell and eukaryotic cells. Prokaryotic cells, as we have seen, where DNA is present in circular manner. It is in cytosol actually. It is present in the cytoplasm. Whereas in eukaryotic cell, the DNA is a linear type. It is present in the nucleus. And another important thing is, here in prokaryotes, we can see only one chromosome, that is circular DNA molecule. Apart from that, we can see an extra chromosome, that is called plasmid. But in eukaryotes, there are number of chromosomes that is very particular to a species. So all the chromosomes are in linear fashion during cell division, but after cell division all these chromosomes are connected and form a chromatin network which is present in the nucleus. In prokaryotic cells, you do not see all the major important organelles like mitochondria, chloroplasts, endoplasmic reticulum, ribosomes. But in eukaryotes, you can see double membrane structures and single membrane structures like chloroplast, mitochondria, nucleus, endoplasmic reticulum, Golgi complex, microbody, etc. Here, the nucleus is not clear because it is not enclosed by nuclear membrane. Hence, the nucleus in the prokaryotes is called nucleoid. But it is very clear as it is enclosed by a membrane. In eukaryotes, it is called nucleus and it is fully developed. As I told just before in the beginning of this slide, prokaryotes are with a single chromosome and eukaryotes are with several chromosomes. That is depending upon the species. And here in prokaryotes, below the cell wall, plasma membrane is present. That plasma membrane is not with the receptors. But in eukaryotes, the plasma membrane is present below the cell wall in case of plant cells, and in case of animal cells, plasma membrane is the outermost layer, and the plasma membrane in eukaryotic organisms or cells are with the special receptors like sterols and carbohydrates, which carry signals from outside to inside the cell and from inside to the outside of the cell. Chemically, prokaryotes contain complex cell wall. It is mainly made up of peptidoglycan. It is somewhat complex and thick. Whereas in eukaryotes, cell wall is present only in the plant cells and fungi. No other eukaryotic organisms show cell wall. Plants means algae, fungi, bryophytes, all would come there. In plants, especially the cell wall is made up of cellulose, whereas in fungi, the cell wall is made up of chitin material. And here in prokaryotes, protein synthesis for construction of proteins, building blocks of an organism. Actually, it shows two phases, as we all know, transcription and translation. Transcription is nothing but formation of mRNA from DNA, whereas translation is nothing but formation of polypeptide chain from mRNA. In case of prokaryotes, these both transcription and translation take place simultaneously in cytoplasm or cytosol. But in eukaryotes, transcription takes place in the nucleus and translation takes place in the cytosol region. Here in prokaryotes, as we have seen in the diagram of prokaryotic cell, flagellum are present. Flagella are present. Flagellum is single form of flagella actually. It is simple, built from two proteins. It shows hook and shaft. Whereas in the flagellum of eukaryotic organisms, it is long, complex, made up of 9+2 arrangement of show 9+2 arrangement. Actually, this 9+2 arrangement is about microtubules. Microtubules arrange in 9+2 pattern. Central to tubules enclosed by 9 microtubules. The type of arrangement is not seen in the flagellum of the prokaryotes. Here in prokaryotes we can see pili and fimbrial-like structures. But in eukaryotes, especially animal cells, show cilia. Instead of pili and fimbria, they show cilia. Here in prokaryotes, genome is called haploid because only one copy of each gene is seen. Whereas in eukaryotes, diploid genome, that is called diploid genome. More than one copy of each gene. Because in here in diploid genome, one set of chromosomes come from father and one set of chromosomes come from mother. Thus, they show two sets of chromosomes, that is called diploid genome or 2x, denoted by here each gene is present more than one copy. As I said in the beginning of this slide, along with the regular DNA or chromosome, prokaryotes also show plasmids. Whereas in eukaryotes, plasmids are not seen commonly, but certain fungi show these plasmids. In prokaryotes, DNA is very compact type. It shows little repetitive sequences, whereas in eukaryotes, large amount of non-coding sequences and repetitive DNA sequences can be seen. In prokaryotes, glycolic calyx cover may be present, whereas in eukaryotes, the cells or organisms which do not have cell walls may show glycocalyx. Here in prokaryotes, ribosomes are small in size, they are 70S type. The subunits of 70S ribosomes are 50S and 30S. Whereas in eukaryotes, ribosomes are large in size, they are called 80S type. The larger subunit of 80S type of ribosomes is 60S and the smaller one is 40S. They are present in both cytoplasm as well as in the nucleus, and also they are present in the mitochondria and chloroplastids. Whereas ribosomes in the prokaryotes are present only in the cytoplasm. Actually, in prokaryotes, as there is only one DNA molecule, it is not associated by or associated with the histone protein, so that it is always active. Whereas in eukaryotes, the histones are always closely associated with the DNA molecule because they are many number in eukaryotes. So all the DNA do not work at the same time. The DNA which are essential or needed at particular time, they would act at particular time, and later on other DNA according to their needs. So the shutting off DNA molecules is done by association of histone proteins. The DNA which are active are free from histone proteins. And here in prokaryotes, there is no cytoskeleton, that is nothing but endoplasmic reticulum. Whereas in eukaryotes, cytoskeleton, actin present in the animal cells, and microtubules are present in plant cells, so that they provide physical support to the cell. In prokaryotes, mycolaginous capsule is present, whereas in eukaryotes, the capsule is not at all present. The regular size of prokaryotes is 0.5 micrometers to 100, whereas the size of eukaryotic cells range 10 to 150 micrometers. Prokaryotes show binary fission. It is a type of asexual reproduction. Mother cell divides and gives two cells. Whereas in eukaryotes, we can see mitosis and meiosis, and they reproduce by sexual methods. Mitosis is a type of division which is seen in the vegetative cells, whereas meiosis is a reduction division which is seen generally in the reproductive cells, so that we will get gametes. So by this video lecture, we have learned what is basically a cell theory, the important points of cell theory, and the important differences between prokaryotic cell and eukaryotic cells. I think you have understood all the details given by me by this video lecture. We'll meet soon with another video lecture. Till then, bye. Have a nice time. Thank you very much.